Hi, everyone. Welcome to 122nd Stanford MedAI Group Exchange session. This week, we have Xiaohan Xing from Stanford University here with us to talk about their work on integrating pathology images and genomics data for cancer grading. Xiaohan is a postdoctoral researcher at Stanford University. She has obtained her PhD from the Chinese University of Hong Kong. She has been extensively involved in research related to medical AI, focusing on disease diagnosis and survival prediction based on medical images, omics data, and the integration of multimodal data. And she was honored with the McKay Young Scientist Award in 2022. Xiaohan, thank you very much for joining us today. Before we start, do you have any preference on how you would like to take questions? Do you have like time breaks in your talk, or you would like to take questions at the end?
Yeah, you can ask questions at any time.
Thank you.
during
In
the talk.
that case, let's try to make this session as interactive as possible. And let me hand it over to Xiaohan.
Okay, thank you for the introduction. Thank you for the invitation. Good afternoon, everyone. My name is Xiaohan Xing, a postdoc researcher at Stanford University. My research focuses on AI for healthcare, and today I will introduce my research about integrating pathology images and genomics data for cancer grading. Precision medicine requires a comprehensive understanding of the disease, especially cancer, which has a very complex disease mechanism. Human bodies go through a series of changes from molecular to phenotype. From the molecular level, they may cause aberrations in gene expression, such as genomics data, transcriptomics, and proteomics data. From the phenotype view, the aberrations can be detected by different types of medical images. As a result, both the medical images and omics data, changes in omics data and the medical images can reflect the disease status and thus are widely used for disease diagnosis and survival prediction. Recently, artificial intelligence has been revolutionizing healthcare. A simplified pipeline is shown here. After collecting multi-modality biomedical data, we can analyze the data characteristics and design efficient AI algorithms to analyze this data. A lot of tasks can be performed: disease diagnosis, survival prediction, drug discovery, and treatment response prediction, and etc. According to the data modalities, my previous research can be classified into 3 categories, including the medical image analysis, omics data analysis, and the integration of pathology slides and the genomic profiles. So this is a brief introduction of my previous research outline. For medical image analysis, my research includes verification on the diagnosis of small lesions on the endoscopy images, and class-guided contrastive learning for the classification of the endoscopy images, and also studied how to train a robust and reliable model with the existence of noisy annotations. And then for the omics data analysis, we developed a multi-level graph network to extract the pathway information, improve the glioma grading and survival time prediction based on the transcriptomics data, and improve the COVID-19 diagnosis based on proteomics data. In the next part, since both medical images and the genomics data can reflect the disease status, so in this direction, we study how to combine the multimodal data and achieve more accurate cancer grading. Today, my talk will focus on the third part. If you are interested in my work on medical image analysis and omics data analysis, you can refer to these papers. You are welcome to email me if you have any questions regarding these 2 directions. So any questions so far? Okay, now let's move to the main topic of this talk. The basic idea of this research direction is that the pathology slides and the genomics data can provide complementary information. So their combination can be expected to achieve more accurate cancer diagnosis. As shown in this figure, the pathology slides at different scales can reflect the cell shape, organization, and interactions among neighboring cells. Thus, they can depict the tumor microenvironment, which is closely related to the cancer grading and the survival time of a patient. On the other hand, The genomics profiles such as gene expression and copy number variation, which can also reflect the status of the tumor progression. Therefore, the morphological information in the pathology slides and the molecular information in the genomic profiles can reflect the disease status from different aspects and can complement each other. Recently, there have been a lot of efforts dedicated to the integration of these two modalities. Challenges we need to consider during algorithm development. The first challenge is modality gap. As the pathology slides are 2D images and the genomics data is 1D vector, these two modalities have a huge gap in their feature space distribution, and there are some redundant and noisy features in each modality. Therefore, how to exploit the cross-modal complementary information while mitigating redundant information. And the second challenge is the modality imbalance. This means the contribution of different modalities are imbalanced. Let's take the glioma grading as an example. If we evaluate the accuracy of these two modalities separately, we can find the accuracy of the pathology slides is about 73%, and the accuracy of the genomics modality is only 65%. Might be dominated by the pathology features and fails to fully exploit the useful information from the genomics modality. Therefore, we need to design a network that can balance the contributions of different modalities and better exploit the useful information from the weak genomics modality. And the third challenge is modality missing. This is very common in clinical practice, especially for the because the genomics data requires high technique and high cost. So not every patient can collect the genomic data in clinical practice. The missing of the genomics data will lead to significant performance drop, as shown here, from 75.7% to 73%. So we need to consider how to improve the prediction accuracy of pathology slides when the genomics. So today I will introduce three of my previous works that aim to tackle these three challenges respectively. For the first challenge, we develop a low-rank constraint-based method to integrate the multi-modal data. Low-rank constraint has been widely used in multi-modal learning and multi-view clustering. This is a picture from the IGCV paper in 2018. This paper low-rank constraint. It is a common practice to construct a tensor by stacking multi-view subspace representations, Z1, Z2, and Zv, as shown here, and regulating the low-rank property of this tensor Z. In this way, different modalities can interact with each other. Thus, the cross-modal consensus and complementary information can be exploited. This method works while clustering when the different views are not there is no huge modality gap between different views. However, we experimentally find this method doesn't work in the fusion of pathology slides and genomic profiles, since a huge modality gap exists between these two modalities. To tackle this challenge, we propose this momentum-enriched multi-modal low-rank constraint. It is called M3L. Instead of imposing low-rank constraints across all individual modalities, we construct a low-rank tensor for each modality separately. As shown here, for each modality, we extract two sets of features with this encoder E1 and this momentum encoder E1 hat. For these two input modalities and the fused modality, we can get 6 features in total. Then for each modality, we construct multi-modal tensor by stacking the similarity matrices of the fused modality and the momentum encoders, and regulate the low-rank property of these tensors Z1, Z2, and Zm. In this way, the fused modality can serve as a bridge between different modalities and provide more consensus information. the momentum encoder, it is updated by the exponential moving average of, thus providing richer complementary information of different training steps.
Hi,
After
Xiaohan, can I ask a question on the last slide?
sure.
So if there is a modality gap, the way you present modality gap is if it's, for example, pathology images, there'll be 2D, and the genomics data, that'll be 1D. So in that case, your encoders for the two modalities, they will have different shapes so that one of them takes a 2D data and shape and, projects it into, let's say, n-dimensional space. And then the second one does the same for a one-dimensional data, right? So they take different size inputs and outputs, same size output. Is that correct?
Actually, the input of the pathology slice is 2D, but in the feature level, it is we extract a 1D feature for this modality. But because these two modalities are very different continuous modalities, so their feature distribution will be very different. Their combination cannot be directly achieved by imposing the low-rank constraint on these two different features.
But would they be in like similar dimensional space?
Yeah. In a feature dimension, they are the
They're
same.
similar.
Yeah.
Okay.
But the distribution will be different.
Distribution.
Yeah.
Makes sense. Thank you very much.
Thank you. Any other questions? And then after constructing the momentum-enriched multi-modal tensor for each modality, we regulate the low-rank property of this tensor by transforming it into the Fourier domain and performing singular value decomposition and shrinkage. In this way, the consensus information among different modalities and different training epochs is captured to achieve better feature representation of the multimodal learning network. For we use the glioma dataset curated from the TCGA database. For the pathology slides, we use the ROI images instead of the whole slide images. The genomics data include 79 features for copy number variation and 1 feature for gene mutation. The dataset is used the same as this paper. The model performance is evaluated by 4 metrics, including accuracy, average precision, and the F1 score of the grade 4. The results show that our method outperforms existing multimodal learning methods by a large margin. Compared with the baseline model proposed by Chen in 2020, our method leads to 3% improvement in accuracy. This is mainly due to our proposed low-rank constraint because it can better exploit the cross-modal complementary information.
Now, we have a question from the audience. I think it's about the ROI. What are the ROIs used for pathology images? Just tumor core tiles?
Because the whole slide image is very large, the resolution is about several sovereigns times several sovereigns, right? So this is the dataset provided by another paper. In that paper, they annotate the region of the tumor and cropped the tumor region as the ROI. Yeah.
Okay,
it's
so
the region
there is
of
like
interest.
manual annotation of the tumor
Yeah,
region.
it's manual annotation. And this annotation can also be achieved automatically. There are some tools already developed by other groups. They can automatically crop the region of interest.
Thank you.
I have questions regarding that future fusions in that previous slides.
Okay.
So, I mean, I'm confused with the way of combining those features. Would you like to explain a little bit? Yeah, this is some future modality. Yes, yeah.
Yeah, it is inspired by the, should I start from here? The
Yeah,
low-rank constraint?
That I understand, but the next slide.
Next slide.
Yes.
Yeah, it is inspired by the low-rank constraint, but if we directly stack different similarity matrices, different modalities, the huge modality gap will constrain our feature representation learning. So in this method, this is the blue box. This blue box, these four similarity matrices form the first low-rank tensor for the pathology modality, and the green box is the rank tensor for the genomics modality. These two are the features for the pathology, and these orange features are for the fused modality, and the greens are for the genomics modality. So we stack the genomics modality with the fused modality to form the low rank tensor for the genomics modality. The fused modality is it can serve as a bridge between different modalities because it contains information from both modalities. So it can, to some extent, reduce the huge modality gap.
Okay, so I was confused with the F because I thought you have another expression in your equation. So maybe okay.
Oh, you mean this F?
F, yeah.
Oh, sorry. This F means the fusion. It is performed by Kronecker product.
Yeah. Okay. Yeah, sure. Thank you.
Okay. Okay. Next slide. Okay. Any questions about the method part? Okay, let's move to the results. Let's continue from here. And we also conducted ablation studies and found the compared with the baseline model here in the first row. Introducing the M3LR constraint leads to gains. And more detailed comparisons reveal that both the fused modality and the momentum fuse contributed to the performance gains, indicating the effectiveness of our method design. And we also, similar as previous works, as shown here, we also use the tensor nuclear norm to measure the low rank property of the multi-modal tensors and find our M3LR constraint can effectively use the TNN, which is the reason for the performance gains. And we also found that with this constraint, not only the multi-modal accuracy is improved for about 3%, accuracy of the pathology branch is also improved from 73 to 75%, and the genomics accuracy is improved for about 5%. And these results indicate that the low-rank tensors can provide information for each modality and suppress the modality-specific noisy features. So this is the introduction of my first work. And questions about this work? Okay, now let's move to the second work. This work aims to tackle the second challenge of pathology genomics data integration. It is the modality imbalance. For the glioma grading task, the accuracy of the pathology modality is much higher than the genomics modality. Thus, the multimodal network might be dominated by the pathology features while having an insufficient exploration of the genomics modality. So to tackle this challenge, we propose a Saliency-aware masking strategy to train the network. Specifically, based on the gradient information, and based on the gradient information, we mask some of the most important features from the input data from both modalities. And then we feed the complete data here and the masked data into the multi-modal network. By enforcing the model to make the same predictions on the complete input data and the masked input data, the model will be forced to explore these but neglected features, especially the features in the weak genomics modality. Therefore, this training strategy will mitigate the modality imbalance issue and balance the contributions of different modalities. Details of the Saliency-aware masking strategy are shown here. We first compute the gradient of the input pixels and the genes as shown here. And because the gradient of the input pixels might be to improve the robustness of the saliency computation on the pathology images, we further divide the images into superpixels and compute the superpixel level saliency scores. By masking the most silent superpixels and genes, we can get the masked input and conduct the masked training that we introduced earlier. Any questions about this method?
Yes, and Zion, I think I'm going to start with one question. What is this erasing function at the end?
Erasing function, because we have generated this mask. For this mask, we set the most important superpixels and genes as 1, and other parts as 0. So after this 1 minus this mask, the most important features will be erased.
Sorry, so I think I'm failing to understand the intuition behind erasing the most important features. Why not highlight those features? Like, shouldn't it be reversed?
Yeah, because we want to keep the consistency between the complete input data and the masked input data. Because except for the most important features, there are some other less they are ignored by the network, because this importance is measured by this silence is measured by the model, right? Yeah, it means this model focus on this part, but if we erase this part, the model will try to find other features that are also very important, but it's ignored in the previous training.
Okay,
so in this way, the model will try to the same prediction based on this complete data and the incomplete data. It will explore by more comprehensive disease-related features.
And do you have to do some kind of like intelligent hyperparameter tuning? Because if you're masking the most important feature,
yeah,
I think in very few scenarios you will get the same like output as you will get with those features. So you have to do like intelligent weighting of this objective. I mean, try to get the output to be the same with and without masking, but don't force the model too much, otherwise it'll probably not
yeah,
learn.
that's a very good question. Actually, in our paper, we performed an ablation study to analyze how many features we need to mask. If we mask too much features,
Yes.
it means the remaining features are not important. If we force the network to focus on the, it will be bad. So we fine-tuned the number of features we try to mask. It is a very important hyperparameter.
That's very interesting. I think we have one more question from the audience. I think it's very interesting. If only pathology model is dominating, why apply masking to both modalities? What's the intuition behind applying the masking to both modalities?
Yeah, that's a very good question. Actually, in our experiments, we all studied if we mask both modalities and if we mask only one modality, and we found masking both modalities achieve better performance. And I guess the reason behind is because we for the pathology slice, although it is the dominating modality, it may focus on. I can show you with the results as shown here. For the pathology slides, you can see from this figure, the baseline KIM, this is the saliency of the baseline model. It may focus on a certain part of the pathology slides, but if we mask this most important part, it will focus on more comprehensive regions. So for this pathology slides, if we mask the most important region, it will also force the network to achieve better exploration of the features in each modality. Although it is the dominating modality, there are also it also contains some important but neglected features.
So intuitively, this kind of masking and erasing, it would just improve the performance, even if you're using just one modality. Let's say you're building a model with just like pathology slides, and with or without this masking, the performance should improve because the model kind of tends to learn comprehensive feature rather than focusing on like very small regions. Is that right?
Yeah, if we only mask one modality, it will also cause improvement to the model compared with the baseline. Masking only pathology modality or masking the genomics modality will also lead to performance gains.
And if you do both of them, you get the most performance improvement.
Yes,
That's interesting. Thank you.
Because both for the stronger and the weaker modality, they both contain some important features that are neglected by the model. So we will force the network to explore the features more comprehensively.
So would you say it's because you're using distillation? This is better when you apply to both modalities?
Sorry, can you
can you hear me?
clear? Yeah, now I can hear.
Okay, sorry about it. So would you say that because you're applying some form of like distillation learning, when you mask like both the modalities, it is better than just one?
You mean diffusion model?
No, distillation. Like if you go back to
Distillation.
yes.
So is it like close to knowledge distillation, that techniques?
Knowledge distillation between these two modalities.
Yeah, if you go back to, I don't know which slide number. Yeah, here. Yes.
This one? Okay.
Yeah. So what is this EMA that like is applied from the masked model to the unmasked?
This EMA means exponential moving average. It is a common mean teacher. It is used in knowledge distillation. Yes, that's right. And we use this. The first model is the EMA model of the second model. So we want to use different models to because the EMA will contain more knowledge than the vanilla model. So we actually this is more robust. The EMA model is more robust than the so we want to keep divergence between these two models. And in this kind of divergence, we also want to get this consistent prediction. It is kind of perturbation to the model, and this is a perturbation to the input. There are two different levels of perturbations.
Okay, thank you.
Okay, thank you.
I had a question. So
Okay.
even after like knowledge distillation, so not knowledge distillation, sorry, even after the masking, how do you know that all the other information that the model is kind of focusing on, those are important and actually will contribute to the model's learning? Like you're already kind of eliminating all the dominant features, but the dominant features might be the more useful features, right? Like the other features might not be like, okay, fine, with these pathology and this data set you're getting really good results, but it might happen in something else. And when you're doing some kind of an external learning or some kind of a data set which is not this one, then this method might not work. So how do you did you do some kind of an external validation or a data set which was different from this?
Yeah, this is a very good question. I think this may happen for some data sets or some other not work because sometimes one modality is dominant and we want the modality to be dominant. It will make more contributions and help the better learning of the entire multimodal network. Yeah, that's true. I believe that's true. And I think the most important unimportant hyperparameter is how many features we need to mask. If we mask if we set the number of superpixels to be masked as 1, I think that is a very rational number. If we set it as 1, it means only 1 superpixel is masked. For especially for this application, it is very common for more than 1 superpixel are very important to this task. I think it depends on the task. For this task, it is because different superpixels and different genes, they both contribute to the disease grading, for the cancer grading. But for other tasks, maybe only one feature or two features that are very important, and other features might be noisy and may damage the multimodal learning. So in that case, this method may fail.
May fail, yeah.
Yeah, that's true.
Yeah.
It depends on the application and the data set.
Exactly. Yeah.
Okay.
Okay.
Thank you. Very good question.
Thank you.
Okay, let's move on to the result part. And we also compared with the multimodal fusion methods and found our method achieves the best results. And we performed the ablation study on the masking strategy. And we found masking the most salient features of both modalities leads to better performance gains, or masking only one modality. And this result is consistent with our assumption, as the relevant features can be more comprehensively explored by masking the dominant features from both modalities. And here shows the Saliency-aware masking strategy can improve the accuracy of both modalities according to these statistics. And then the visualization of the attention maps. Here shows the attention maps of the baseline model and our model. And we can see that the baseline model may only focus on small region of the image. And our method leads to more comprehensive exploration of the disease-related features. It has a better coverage of the most important features. So this may explain why our method will lead to performance gains. Okay. Second work. Any other questions about this work? Okay. Now let's move to the third project. It is about the knowledge distillation, but it aims to tackle the third challenge in the pathologic fusion, to tackle the missing modality issue. As we have mentioned earlier, genomics data is not always available in clinical practice due to the technical requirements and high cost. Multi-modal network cannot be used due to the missing modality issue. So to tackle this issue, we train a unimodal network by distilling knowledge from the well-trained multimodal teacher. To maximize the knowledge transfer, there are two streams of knowledge distillation methods that we can refer to. The first one is devising a novel knowledge, and the second is assembling multiple knowledge. From these two aspects, frequency supervised contrastive distillation and gradient-guided knowledge modulation modules. In the next slide, I will introduce the details of these two modules. So the first module is inspired by contrastive knowledge distillation. Our DSCD module aligns the feature distributions of the teacher and the student model by contrasting samples. But the difference is that the existing instance contrastive, they regard samples from the same class as negative pairs and push them apart, such as this sample and this sample. They should be the same class. They should be positive pairs, but they are regarded as negative pairs. And this operation will damage the class semantics and degrade the classification performance. So to maintain the class semantics, we define all samples from the same class as positive candidates. The top number KP informative samples as positive pairs, such as for this anchor point, these two blue rectangulars, they are all positive candidates, and this one is selected as positive pair. And the informativeness is defined by this equation. If the distance between the query image and a positive candidate is closer in the teacher model than in the student model, this candidate is selected as a positive pair because we think it is better classified in the teacher model. And then we sample number kn negative pairs from different classes, such as these two negative pairs. With these positive and negative pairs, our DSCD module can distill richer relational knowledge and maintain the class semantics of the student model. And the second module is the gradient-guided knowledge modulation. Several previous studies indicate that the integration of multiple teachers will provide richer knowledge and nurture better students. So except for the multimodal teacher TM, as shown here, we introduce a unimodal mean teacher TU. This model is the exponential moving average of the student model. Therefore, it allows the retrospection of previous lessons and provides robust intramodality knowledge. Similar to the multimodal teacher, this unimodal mean teacher can also provide knowledge where the KL divergence and our proposed DSCD module. So there are five knowledge in total, including the DSCD and the KL divergence from the multimodal teacher, the KL loss from the unimodal mean teacher, and also the cross-entropy loss supervised by the ground truth. So we can construct a knowledge bank with these 5 types of knowledge. Considering that different knowledge is not equally reliable and may contradict each other, our GKM module recalibrates the contributions of different knowledge based on their agreement in the gradient space. Way the contribution of unreliable knowledge will be reduced, and the knowledge bank can provide more reliable guidance to improve the performance of the unimodal method, of the unimodal student.
Question over here.
Sure.
Yeah, sorry. I mean, probably I'm not understanding it well, but this knowledge bank, when you are kind of combining all these knowledges from different modalities, is there any redundancy in this knowledge bank that you are constructing, or is it like different views of the same of the data set?
Sorry.
In the knowledge bank that you are creating, is there any redundancy of the features?
You mean this
knowledge bank? Could you repeat once more?
Oh, yeah. This knowledge bank, it contains the knowledge distilled from different sources, from different teachers. And this multimodal teacher, it provides 2 different knowledges, including the KL divergence, KL loss. It regulates the consistency between the predictions of the multimodal teacher and the unimodal student. And our proposed DSCD module, it aligns the Fisher distributions of the teacher and student model. And similarly, the unimodal mean teacher can also provide these 2 types of knowledge. And this CE means the cross-entropy loss supervised by the ground. There are 5 types of knowledge in total. And this idea is inspired by another work. In that work, they point out that the integration of multiple knowledge will improve the performance of the student model. They integrate different teacher models. So in this work, we integrate different knowledge, but we also contribution of different models might be different, and some knowledge might be harmful. So we reduce that the contribution of the harmful knowledge and emphasize the contribution of the important knowledge.
We have one question from the audience: how to determine the regularizer parameters, lambda 1 to lambda 4? So yes.
Yeah, this parameter is decided by this equation. We measure the similarity between the for example, for the there are 5 knowledge. So except for this G example, there are five knowledge with different directions and different strengths, and then we measure the similarity between different knowledge. For example, for the G2, we measure the similarity between G2 and G1, G2 and G3, G2 and GCE, and we get 4 similarity matrices, similarity value, and we compute, we sum the similarity value of different knowledge, and then we, if the similarity is larger than a threshold, then we set the reliability of that knowledge as 1, Otherwise, it is set as 0. The idea behind is because we think different, if a knowledge is reliable, it should be consistent with most of the knowledge. So we measure the similarity between different knowledge to decide whether it is reliable. Okay, any other questions? Okay, thank you. Let's and then we, in the experiments, we evaluate the unimodal network with only pathology slides available for inference. It is a baseline model. And the baseline model is trained with the pathology slides only. And the other comparison methods are trained by distilling knowledge from the multi-modal teacher to the unimodal student with different knowledge distillation methods. And from this table, all of the knowledge distillation methods outperform the unimodal baseline. This indicates that the knowledge distillation from the multimodal network can effectively improve the unimodal diagnosis. And more importantly, our method enables more effective knowledge transfer than other knowledge distillation methods, and is very close to the multimodal teacher. It is the upper bound. This means that for some countries and regions with limited memories, if satisfactory diagnosis can be achieved even if the genomics data is missing. So this is the introduction of the third work. Any questions about this work?
I think I have one question here. It's a very interesting thought that if from another region of the world, if the genomics data is not available, maybe we can just use the pathology data.
Yeah.
But have you done some kind of external validation? Because like, you know, medical AI models, they have a lot of generalization. So something that works on this population may not work on another population. So do you think like that the combined, the unified teacher that you're talking about, if it is trained on one population, it has a like, what's your intuition? Does it have a good chance of generalizing to another population?
Yeah, very good question. In work, we didn't perform external evaluation. We only evaluated on the same data set. We performed the cross validation on one data set. But in the future work, I'm trying to collecting more data from different sources and evaluate the model on different populations. Yeah, that is a part of my future work.
That'll be very interesting. Thank you.
Yeah, yes. That will evaluate the generalization of our model and clinical applications. Yeah, that's very important. Okay, so let's summarize this talk. In summary, to improve the cancer grading based on the integration of pathology images and the genomics data, we developed 3 algorithms to tackle the 3 challenges. First, to bridge the modality gap between the heterogeneous modalities, we developed a momentum-enriched low-rank tensor explore the cross-modal complementary information and reduce the modality-specific noise. And to balance the modality contributions, we developed a Saliency-aware masking strategy to explore the disease-related features from the weak modality. And to tackle the missing modality issue, we propose a novel knowledge distillation framework with the DSCD and GKM modules. And the experiments show that our method performs both multimodal and unimodal diagnosis. In the future work, I will continue my research in multimodal-based AI for healthcare. I plan to conduct research on these 3 directions. First, we can include more modalities, such as spatial omics, proteomics, and medical reports, and etc. These different modalities will provide complementary information and pave the way to precision medicine. And second, recently, models are gaining popularity due to their ability to understand different data modalities and quickly adapt to various downstream tasks. Therefore, it is very promising to integrate the knowledge from foundation models to facilitate our multimodal task. And third, instead of working on disease diagnosis, I will explore the potential of AI models in more challenging tasks beyond the doctor's capabilities, such as mechanism discovery and treatment response prediction. So if you are interested in these research topics, you are welcome to email me and we can discuss about the opportunities to for collaboration. So this is my report. Thanks for your listening.
Thank you very much. Thank you very much, Xiaohan. It's very interesting talk. Before we open the floor for questions, I think we should all give our speaker a virtual round of applause. And then if any of our audience have any questions, they can just ask.
Yeah, I just have a quick question.
Mm-hmm.
For the feature masking, are you masking the same number of features for each modality, or are you masking more of the pathology features just because it's the dominant modality?
There are two separate hyperparameters. The number of features we mask and in the pathology modality and the genomics modality. These two hyperparameters are separately fine-tuned, and we according to my I remember the number of superpixels we mask is maybe 5, and the number of genes we mask is 1. So actually, we mask more features from the pathology modality because it is dominant.
Right. Okay, that makes sense. Thank you.
Thank you. Any other questions?
I think I have one last question. Basically, how do you see the application of your work? Because the methods you've been proposing, they're really kind of modality agnostic. Like you can take the same approach and you can apply it to vision and text, and then maybe vision and tabular data. So have you any thoughts on like expanding into different modalities?
You mean extending these methods to other modalities like the tabular data, text?
Exactly. Yes.
Yeah, I think because the method we proposed is not targeted as a specific modality. We study the general multi-modal fusion or the missing modality techniques. So it can be, I think it can be extended to other modalities. We just need to use different encoders to extract features from different modalities. I think it can be easily extended to other applications.
That's very interesting.
And integrating more modalities is also my research interest in the future work.
I think if we have no more questions from the audience, let's just give our speaker another round of applause. And if anybody has any questions later, we can put you in touch with the speaker.
Okay. Thank you.
Thank you very much. Thank you very much, everyone, for joining. And thank you, Xiaohan.
Thank you, everyone.